is Kavya Tiwari. I'm currently a second year student at Banaras Hindu University. I'm pursuing my masters in botany. So today I'll be teaching you photosynthesis, basically the light reaction of photosynthesis. So we will just revise what photosynthesis is. Basically plants make their own food in presence of light with the help of carbon dioxide, water and pigments. So and to form sugar, which is glucose, and it releases oxygen into the atmosphere. So we will study the light reaction part basically. So basically photosynthesis is divided into the light reaction and dark reaction. Two phases it has been divided. In the light reaction, the products formed are ATP and NADPH2. And in the dark reaction, sugar is formed, glucose as I have said. The location of light reaction is thylakoid membrane and the location of dark reaction is stroma. Basically, dark reaction doesn't mean that it takes place in dark. In the light reaction takes place in light. It forms ATP and NADPH2, and by using this ATP and NADPH2, this sugar molecule is formed. So we cannot say that dark reaction can take place in dark, because it depends on the product of light reaction, that is ATP and NADPH2. So light reaction is also called the Hill's reaction because it was discovered by the scientist Hills, and dark reaction is called Blackman reaction because it was discovered by Blackman. Basically, the light reaction is also called the photophosphorylation step. Photophosphorylation. Why photophosphorylation? Because we will break this word and understand. Photo means light. That means it is taking place in presence of light. And phosphorylation in this context means the formation of ATP. How this ATP is formed? This ATP is formed by addition of phosphorus to ADP. So basically we are doing what? We are phosphorylating this ADP to form ATP, and it is taking place in presence of light. That is why we are calling it photophosphorylation. Now we will discuss how photophosphorylation is divided into two phases. So now, as I have said, we will discuss the photophosphorylation. It is of two types, non-cyclic and cyclic. In case of non-cyclic photophosphorylation, electron flow is non-cyclic. We will see how. And cyclic, electron flow is cyclic. So basically, non-cyclic photophosphorylation, both PS1 and PS2 is involved. In case of cyclic photophosphorylation, only PS1 is involved. We'll quickly discuss about photosystems. So there are two types of photosystem. Why we are calling it system? Because they have more than one component. That is why we are calling it system. So, photosystem 1 and photosystem 2. Photosystem consists of basically the reaction center plus light harvesting complex. For light harvesting complex, I am writing LHC. So in the reaction center comprises of one molecule of chlorophyll A. And for chlorophyll A, I am writing CHA. And in the light harvesting complexes, there are hundreds of pigments, which are bounded with protein. So basically the main component is reaction center, but light harvesting complexes are very important because it increases the efficiency of photosynthesis. So here PS1 and 2 both are involved, here PS1 is involved. Now we will see that in case of non-cyclic photophosphorylation, there is an involvement of OEEC, which is oxygen in evolving complex. We will see how it will give electron in the non-cyclic photophosphorylation. But in case of cyclic photophosphorylation, there is no oxygen evolving complex. And in case of non-cyclic photophosphorylation, there is production of ATP plus NADPH2. But in cyclic, there is only production of ATP. No NADPH2 is produced. That means here two enzymes are present, which is the ATP synthase for the formation of ATP, and NADPH2, and the enzyme NADP reductase for the formation of NADPH2. And here only ATP synthase is present. So now we will discuss about the non-cyclic photophosphorylation. So we all know that ultimate source of light energy is sun. So from sun the radiation will strike the PS2. Now firstly we need to learn that PS2 and PS1, two photosystems are involved in non-cyclic photophosphorylation. These two has the same reaction center and light harvesting complexes. The only difference is it absorbs the wavelength of 680 nanometer and it has an absorption maxima of 700 nanometer. So now we will start. From sun, the radiation, the light energy will strike the PS2. Then there is excitation of the electron from PS2. This electron will move to a higher energy state. Then this electron will be passed on to pheophytin. These all are electron carriers. So these electrons will be passed on to pheophytin. Then from pheophytin it is transferred to plastoquinone. Then from plastoquinone, cytochrome B6F. And from cytochrome B6F we have a mobile carrier. Why mobile? Because it can move. And it can move and it can just pass its electron to PS1. One more thing, that these electron carriers basically are arranged in an order of increasing order of redox potential or reducing potential. Basically, what is reducing potential? It is a capacity to accept electron. So we can say that pheophytin has low redox potential, then plastoquinone, then plastoquinone has low redox potential, then cytochrome B6F. So likewise, these will take place. And this sun's energy will also excite PS1. It will get excited by PS1 and it will also get the electron from plastoquinone. Now, after getting this, it will also be excited. It will move to its excited state. Then it will move through a series of electron carriers. For example, firstly the iron sulfur cluster, then the ferredoxin, and finally the FNR. What is FNR? This is ferredoxin NADP reductase. It will reduce the NADP plus to NADPH2, and this is how NADPH2 is formed in light reaction. Also there is production of ATP. The formation of ATP is dealt by a different concept which is called the chemiosmosis theory, which can be discussed later. Now if you see here, electrons is getting excited, it is flowing through a series of electron carriers, and then finally it goes to FNR, then NADH2 is formed. But who will fill the gap of electron here? There should be someone. So here one more complex is involved which is called the oxygen evolving complex. The oxygen evolving complex will basically split the water. That is why it is called this process is called the splitting of water. Why splitting of water is taking place? To give 2H plus 2 electron plus half O2. So what is happening here? This electron, this gap which has been created here will be filled by this electron. And this oxygen will be released into the atmosphere. So this is about the non-cyclic photophosphorylation. Now we will quickly discuss about the cyclic photophosphorylation. In case of cyclic photophosphorylation, there is involvement of only PS1. So from here, only the photosynthetically active radiation will strike the PS1, then electrons will get excited. It will be transferred to FES, which is iron sulfur cluster, and from iron sulfur cluster it will be transferred to ferredoxin. From ferredoxin it will come to cytochrome B6F, then it will come to mobile carrier plastocyanin, and then plastocyanin it will come back to PS1. So here we can see that the electron which has been excited here is coming back to its original position, and then this process is going on. So we can call it cyclic. As we can see, the electron flow is cyclic. Here there is also formation of ATP. ATP is formed. The only difference is here no NADPH2 is formed. So there is no FNR. And in case of PS1, there is no oxygen evolving complex. So O2 is not evolved. That is why, as the electron flow is cyclic here, so it is called cyclic photophosphorylation. So this brings me to the end of this lecture. It was pleasure teaching you all. Thank you.